So it's my pleasure today to introduce our speaker, Professor David Wilson. Professor Wilson was one of the premier investigators in cellulases, and he began his educational career at Harvard, where he got his bachelor's degree. He then went on to get his PhD at Stanford Medical School, and then did a postdoc at Johns Hopkins. After that, he joined the faculty at Cornell University in 1967, where he remains today. At Cornell, his research is focused primarily on the mechanisms of carbohydrate-active enzymes from the cellular bacterium Thermobifida fusca. His work has sought to fully characterize the molecular mechanisms of the polysaccharide breakdown of this organism and to engineer improved enzymes with the primary goal of more effective cell wall hydrolysis. And with that, Professor Wilson.
So it's a great pleasure for me to return to Berkeley. I actually did a summer NSF with Melvin Calvin, who this building is named for, back in 1960, I believe it was. So it was quite a while ago, but it was a very interesting summer, and I learned a lot and enjoyed it. And I always thought Berkeley has a really beautiful campus. And when I was a graduate student at Stanford, I also came up here a moderate number of times because my wife was getting a PhD in economics at Berkeley, and so I used to visit. And so it's nice to be back and see that Sproul Hall is still there and the fountain is still there, though everything else seems to have changed quite a bit. So as has been stated, I've been studying cellulases now for quite a while, and they have become my favorite enzyme. And I think cellulases are interesting for two reasons. The first is a scientific one, that in point of fact, enzymes that attack insoluble substrates like cellulases do are, now why is this doing this? What do I want? Oops. Doesn't seem to have the...
Go on left. Yeah, click the screen button.
Yeah, I'm sorry. Oh, that one. Okay, there we go. Okay, thanks. Because there actually are a lot of challenges that an enzyme that faces an insoluble substrate has that are completely different from what a normal enzyme. The most important one being that it's the enzyme that has to diffuse to the substrate rather than the substrate diffusing in the enzyme. And the first interesting thing that came out of study of cellulases, they were in fact the first of this type of enzyme to be really extensively studied, is that in addition to a catalytic domain, you usually have a substrate binding domain. And when they were originally found on cellulases, they were called cellulose binding domains, but because they got found on so many other enzymes that involved in carbohydrate metabolism, they changed it to carbohydrate binding module. And as I say, as far as I know, almost every enzyme that degrades an insoluble substrate, and they're not just polysaccharides, but proteases and polyhydroxybuterases have substrate binding domains. And the reason, presumably, is that you can actually anchor the enzyme to the substrate, and so most of the time the catalytic domain is right close and can find the substrate, whereas if it's floating around randomly in solution, a lot of the time it won't be near the substrate. But there's still actually a lot of argument and discussion about whether CBMs may do other things to the substrate besides anchoring the enzyme, and I think that's still an open question, though I'm beginning to favor that maybe they really do it. The other interesting thing about cellulases, which is discussed in the first slide, is that they really are the most diverse enzyme that attack a single substrate. And so by sequence similarity, there are 11 different cellulase families, and probably one or two more have been discovered since I made this slide. And there are 7 different protein folds. And that's really unusual, because every single cellulase cleaves the beta-1,4 linkage in between 2 glucose residues. So, and as one of the reasons why there's so many different cellulases is because, in fact, no cellulitic organism that I'm aware of degrades cellulose with a single cellulase. They all have multiple cellulases. And in fact, synergism between cellulases is a key part of crystalline cellulose hydrolysis. And it's really important that it's only on crystalline cellulose that you see synergism. At least I do it. Other people report it on other things, but in my experiments, I can never see it unless I'm working with crystalline cellulose. And the reason I believe is that crystalline cellulose hydrolysis is very slow because the enzyme has difficulty finding a segment of a cellulose chain that it can put into its active site. And in fact, I believe very strongly that the rate-limiting step for crystalline cellulose hydrolysis is not bond cleavage, but is in fact the placement of a strand in the active site. And I would argue that at this time we have no idea how the enzymes do that. And so there's really a lot still to be learned about cellulases, and that's been the goal of research in my lab for some time, is to try and understand how that step works. Now, when you look at a given family, they all have the same structure, and they all utilize the same stereochemical mechanism. So there's two mechanisms by which cellulases catalyze hydrolysis that differ in the stereochemistry of the anomeric carbon after hydrolysis, and one it's inverted and one it's retaining. And it's interesting that even though you have these conserved properties in families, you also can have fairly big differences. So there are two families that have both exocellulases and endocellulase. So the same basic protein fold can give you either an endo and exocellulase. And there's also a new type of cellulase that we found in T. fusca that's been shown in almost all bacteria, and that's a processive endoglucanase. And again, in family 9, where that enzyme is found, you have both regular endoglucanases and processive endoglucanases. The other reason that cellulases are interesting, the reason that so many of you are here at the seminar, is because, in fact, people are looking at using cellulases to make biofuels, and this has suddenly become of significant interest, and we'll see how long it lasts, whether it's another flash in the pan like in the 70s, or if this time we'll actually succeed in getting a process that works and it'll become a regular part of our economy. I'm obviously hoping the last is true, but I'm certainly not willing to bet my large sums of money on it happening. But I think it's conceivable. If society could behave rationally, I think it would be a good thing to do. So it turns out there actually are multiple ways in which microorganisms break down cellulose. And the way I am studying, and most commercial cellulases are, is what aerobic microorganisms do, where they actually have a set of individual cellulases, each one of which has its own cellulose binding domain on it, or carbohydrate binding modules, I'm supposed to call them. And these enzymes synergize with each other and catalyze the hydrolysis of the substrate. Now, anaerobic microorganisms, some, not all, have another mechanism in which they actually have large particles that contain many enzymes, including multiple cellulases, called cellulosomes. And in some way that I still don't understand, the interaction between these multiple enzymes on a particle somehow compensates for the lack of access to cellulose surface that the huge particle is going to have that won't fit into small pores and cracks and so on, where much of the surface area is. And they actually are very effective at degrading cellulose. And what I think is quite interesting is if you actually look at the cellulases on cellulosomes, they come from the same families as the ones that are in free enzymes in most cases. One interesting exception is that there are two different exocellulases in many mixtures, not all mixtures. And the reason there are two is because there's two ends on a cellulose chain, and they're different cellulases that attack each end. They're very specific. A given exocellulase only attacks one end. And the enzyme that attacks the non-reducing end, which is a family 6 exocellulase, has never been seen in anaerobic microorganisms. Instead, they actually have one of these processive endoglucanase, which also attacks the non-reducing end. Now, when you get into looking at genomes and look at the cellulases in there, you can see that for both the free cellulases, like T. fusca, this is my favorite organism, that actually has 6 cellulases. There's one extra one, a family 5 one that was discovered later that we never saw when we purified the enzymes from the organism grown on cellulose, even though it's clearly present in the genome, and it is expressed. And interestingly enough, it has a different cellulose binding domain. So every single one of these cellulases that are produced when T. fusca grows on cellulose are family 2 CBMs. And this enzyme has a family 3 CBM. And we don't know what that means, but it suggests to me that it's doing something different, both because it doesn't come out when you grow the organism on cellulose and that it has a different CBM on it. Now, the classical cellulosomal organism is Clostridium thermocellum. And just as T. fusca has 2 exocellulases and a processive end of glucanase, Clostridium thermocellum also has the same exocellulase for the reducing end, but as you see, it doesn't have any family 6 enzymes, but it does have 3 copies of this processive endoglucanase. And another Rumencoccus albus is another cellulosome. Again, it has the 2 processive enzymes. So in both the cellulosomal system and the free enzyme system, exocellulases are clearly very important, or processive cellulases are very important, as well as the CBM. So on the cellulosome, the carbohydrate bonding module is present in the cellulosomal scaffolding protein. It's not on the individual enzymes, but they still bind tightly to cellulose through a CBM, so it is an important part of the thing. If you look at these 2 organisms, Cytophaga hutchinsonii was an organism that DOE sequenced as part of their cellulase project, and I was on the committee, and I protested strongly that nobody had studied these organisms, and it would be much better to do another organism where we knew something about the cellulases, but the microbiologists outweighed the cellulase people, so this is a very interesting organism, and so it got sequenced. And it actually turns out it really is interesting because it clearly is using a totally different mechanism. It doesn't make a single processive cellulase, and almost all of the other cellulases it makes don't have CBMs on them. So it has 2 dramatic differences from the other mechanisms. And what's even more fascinating to me is this is an aerobic microorganism, and Fibrobacter is an anaerobic, and it also has the same, not exactly the same set, but it has the same differences. No processive cellulases and very few CBMs on its enzymes. So it's clear that there are at least 3 mechanisms. And if you look at this final organism that's been pretty extensively studied very recently, this Saccharophagous degradans, it's even more bizarre to me in a sense in that it's got 1 processive endocellulase, which is a totally different type than what we see in the family 9. This is family 5. In fact, most of its cellulases are in family 5. And how you can get an organism with a whole bunch of similar cellulases to degrade cellulose seems to me a really fascinating question that hopefully will get resolved at some point. So this is T. fusca. And it really is an unusual bacteria because it looks just like a fungus to me, except of course it's much smaller in diameter, and it really is a prokaryote. But it's very filamentous. It makes spores like fungi. It breaks down plant cell walls like fungi with the same type of enzymes that fungi use. But it's actually much easier to study. And there are 3 reasons that I sort of decided to work with it and that I think make it easier to study. One is that it actually has the simplest set of cellulases because it only makes 6. Most organisms make even more than that. Secondly, it's partially thermophilic, 50 degrees, 55 degrees it'll grow, and its cellulases are very stable, and that's always nice when you're working with organisms. And it turns out that I didn't realize it at the time, but it's really good at crystallizing its enzymes. So we were able to get really good structures for the cellulases. And in fact, the first cellulase we worked with, the cell 6A catalytic domain, it turned out to give resolution to better than 1 angstrom. And this was the first 30,000 molecular weight protein done that had that kind of resolution. And so obviously my crystallographer was delighted to work with me on other cellulases. And then really I'm always getting requests from crystallographers when they find that T. fusca has one of their enzymes in it because it has been so good at giving crystal structures. So I would guess that it has as many as almost any organism out there, except maybe E. coli or something. So it is a filamentous bacteria, as I say. It's an actinomycete, or officially now they're called high GC gram variable. And it is a major degrader. It's been isolated a number of times. It seems to be a very constant organism. I mean, there are slight differences in some of the isolates, but they're very similar in the set of cellulases they make. So I find that interesting because if you look at anaerobic bacteria, it seems like there's huge variations in the cellulases among different anaerobes of, quote, the same species. But here in this either it's just a growth artifact, since it's so hard to grow many organisms, maybe the T. fusca ones, the only ones that grow are the ones that are like this. I don't know. It'd actually be interesting to do some fish or something and see if there are non-growing ones, that it is more variable than I think. So these are the proteins that we have been studying that are involved. So in addition to the 6 cellulases, which, as I told you, everyone has a CBM on it, and they include 4 different families. There's a cell 5A, which is an endocellulase, cell 6A, another endocellulase. And you can see that these endocellulases are very similar on crystalline cellulose, either filter paper or bacteria, but quite different in their relative activity on other substrates. And then there's cell 6B, which is the exocellulase that attacks the non-reducing end. And you can see right away that exocellulases are terrible on CMC, and that's because they're attacking an end, and as soon as they hit a modified residue, they can't go any further and can't hydrolyze it. So they're limited to the very few residues that are unmodified at the end they attack. And this enzyme has some activity on crystalline cellulose. The other exocellulase, the family 48, that attacks the reducing end, is in fact a terrible enzyme by itself. And yet, as I'll show you, it's very important synergistically. And the 2 exocellulases make up about 70% and are made in roughly equal amounts. So each one's about 30–35% of the total cellulases produced by T. fusca, showing how important the exocellulases are to this organism. And then there's this cell 9A that's the processive endoglucanase. You can see it has good activity on CMC, and in fact, it's the best enzyme I know of on crystalline cellulose. It's got a much 3 times higher activity than any of our other enzymes, and it's much better than CBH1, probably 10 or 20 times the activity of CBH1. And then this really curious to me cell 9B enzyme, which has 2 CBMs on it, like the cell 9A, but they seem to both be binding CBMs. They're not changing the property. It has the highest activity on CMC and is the worst on crystalline cellulose of any of our endocellulases, and I really don't know what it does. And then the last 2 proteins I'll discuss a little about are really interesting. So E78 are very abundant. They're family 33 CBMs, if some of you have heard of them. These were just recently, so they originally found as chitin binding domains. That's what they actually, the first one was a 3 from marcescens that definitely stimulated chitin hydrolysis quite significantly. These enzymes definitely bind to cellulose and to chitin, and we've actually shown Felix Mosier, who's here now, did this work with me as an undergraduate, that they will hydrolyze, they will stimulate chitin hydrolysis. So even though in T. fusca won't degrade chitin doesn't grow on chitin, they clearly are functioning in cellulose hydrolysis, and we've shown they do stimulate cellulose hydrolysis weakly. And then the recent paper from the group that studies this protein showed that in fact it's not a binding domain, it's an oxidoreductase enzyme that specifically cleaves crystalline chitin. And since ours will stimulate chitin hydrolysis, we suspect that it's carrying out oxidative cleavage of cellulose, but that still remains to be proven. But we have shown that, and I'll show you that data soon, that the ascorbate metal ion stimulation we do see in the cellulose hydrolysis with these proteins, consistent with what the other group reported. So this is some of the information I just told you about the family 33 CBMs and the fact that magnesium and ascorbate were shown to stimulate chitin hydrolysis by this protein. And this is a single experiment. We're just beginning to try and check this out and show that we're really getting oxidative cleavage by these proteins. But you can see that with the ascorbate and magnesium, we get a much higher stimulation than we do with the protein alone. This is the activity of just the enzyme. So this is cell 6A, our endocellulase, in the presence of the cell 7. So the reason there are 2 proteins is one is just the family 33 BM. The other, interestingly, is a family 33 domain with a family 2 CBM on it. So again, now that it's an enzyme, it makes sense that it would have a cellulose binding domain on it. And the interesting question is why there's a form that's clearly, it isn't a degradation product of E8. It's a different gene, different family 33 binding domain that doesn't have any CBM on it. So to try and get at one of the experiments I've always wanted to do, which would be to knock out individual enzymes and ask how that affects the overall crude cellulase activity, I realized that I have fairly specific antibodies against each protein, and so we've started doing that experiment. So this is the activity of T-fusca crude all by itself, adding different amounts of the crude, looking at the amount of hydrolysis. This is what happens if you add a control rabbit adisera before you've injected any of the proteins. You actually see a slight stimulation because you're putting a huge amount of protein in there that helps protect the enzyme, presumably a little bit during our long overnight incubations to get hydrolysis of a substrate like crystalline cellulose. And what's interesting, so this is the family 74 antibody, and the point is this is only against the 74. So E8, which is here, so E8 antibody doesn't seem to do anything, but we actually see something with the one, and you'd think there's two proteins. I would have thought I would have had to knock them both out to see an effect on the activity. And it isn't a big effect, but it's definitely real. So the protein is playing a role in the activity of the overcrude. This is the exocellulase that attacks the non-reducing end. It's the most active of our exocellulases, and we're taking that one enzyme out, we're taking out more than half the activity of the crude. So the synergism really is important. I mean, if you look in terms of micromoles per minute per milligram of activity, this is 10 times what the enzyme does by itself in terms of, so it's really doing a lot. And this is that other exocellulase that really had no activity by itself, and it isn't as important to the mixture, but it's almost taking out half the activity when I take it away. So it really, the exocellulases really are key enzymes and have an important role. And again, another thing I find really curious, so this is the E5. I told you I have two endocellulases that are almost equally. The one doesn't do very much when I take it out. The other does quite a lot. The cell 6A one does quite a lot. And again, I would have argued that I ought to have to take both of them out to knock the activity down because, in making a synergistic mixture, either one works, and putting them both together isn't any better than having either one. So there really are some interesting things to be learned here, and we have a lot more work to do in terms of, doing some of the double antibody experiments and really checking the specificity. This one, for example, we've checked very carefully, and I can add the purified enzyme back and get all the activity back in the mixture. So I know it's only taking out the one component, but I need to do that with some of the others. Okay, so now I want to talk about a slightly different story, and that is in addition to the enzyme I showed you that are directly involved in cellulose hydrolysis with pure cellulose, there are three enzymes that T-fusca produces that have family 2 CBMs on them, but they're enzymes that hydrolyze hemicellulose. And they're clearly induced by growth on cellulose. And as I say, they have cellulose binding domains on them, not xylan binding domains or mannan binding domains or xyloglucan binding domains. So we actually did this enzyme in the family 74 because at the time we started, we saw there was one in the genome. We hadn't studied it before. We thought we'd look and see what it did, and we thought it would be a cellulase because that was called a cellulase family. But in fact, it's a xyloglucanase. You can see it's got a very high activity, much higher than any of my cellulases, 189 micromoles per minute per micromole, whereas I had 2 or 5 for the cellulose hydrolysis. It has very low activity on CMC and swollen and none on any other substrate we tested. Now, xyloglucan is a very interesting polymer because it's actually cellulose. It's beta-1,4-linked glucose. But if you take 4 adjacent glucoses, 3 of them will have an alpha-1,6 linked xylose residue sticking out of it. And then some of those can be modified further. And so when we hydrolyze it with this enzyme, we actually get 3 bands on thin layer. So here's the xyloglucan. It doesn't move at all on the thin layer. This enzyme is so active that everything has been completely hydrolyzed to soluble products, and we get 3 different ones because of the different modifications. And this enzyme clips only at the unmodified glucose. And so, what's fascinating to me is that we could actually test the idea that this enzyme's role is to allow cellulases to access cellulose by taking the organism that makes bacterial cellulose and growing it in the presence of xyloglucan. And you can see the product we get out isn't hydrolyzed by a mixture of our pure cellulases. I mean, I wouldn't have predicted it would work that well, that you could actually get all the cellulose coated with xyloglucan so the enzyme couldn't attack it. This is the same substrate after we add the purified xyloglucanase to the other enzymes. And it's degraded just as well as is the bacterial cellulose by our free enzyme mix. So we have clear evidence that xyloglucan can cover up cellulose and that adding this enzyme to the mixture of our cellulose allows our cellulase to access it. And it turns out that T. fusca does not grow on xyloglucan. Even though it completely breaks it down to soluble sugars, it won't grow on it. So the only reason to have this enzyme is for this purpose. And since we also have a xylanase and a mannanase, I strongly suspect that their role is the same. They're involved in, on a real plant cell wall, getting rid of the other polymers. And you can see that if you really want to uncover cellulose, you really want to direct your enzyme to bind to the cellulose, not to the polymer, because you don't, if polymer isn't near cellulose, you don't want to break it down. It's only the xylan or the xyloglucan or the mannan mannose that's close to the, mannan that's close to the cellulose you need to get rid of. So it actually makes a lot of sense to me, though again, it's one of those things I never would have predicted ahead of time. All right, so now I'm getting to talk about real cellulases and the work we're doing fairly recently, looking at site-directed mutagenesis to try and understand both the actual cleavage mechanism and, more importantly, trying to find these residues that might function in this rate-limiting step of putting the strand into the active site. So family 6, as I say, it's an important family because all the exocellulases that attack the non-reducing end are in this family, and it also has endocellulases, and it's found in both bacterial and fungal systems, even though it so far hasn't been found in archaea. And so this is my plug about structures. So we were able to do, this is the structure we got for the endocellulase cell 6A from T. fusca, which at the time was the 5th cellulase structure to be determined. And as I say, it had this really high resolution. And as expected, it had an open active site cleft. It was the first endocellulase in this family. The very first cellulase structure was T. reesii cell 6A, which is an exocellulase. And it has its active site in the tunnel. Now, these don't look like the same protein fold, but they actually are. And the difference in accessibility is due to the loops that cover the active site cleft in this molecule are longer, but more importantly, they're folded in to cover the active site, where here they're folded out. So there actually are significant loops in both sides of this cleft, but they're folded away in this enzyme and folded together to interact and form a tunnel. And every single exocellulase that's been studied has its active site in the tunnel. So there are 3 families. They're totally different structures. They all have an active site tunnel. And again, if exocellulases really do what they're supposed to do, if they're processive enzymes cleaving cell bios one residue at one molecule at a time off the end of the chain, it makes a lot of sense to have your strand stuck in a tunnel. It'll be harder for it to fall off the substrate, and they'll be more likely to function processively. And nature has clearly done that with cellulases. So this is so we actually have a real structure for cell 6B that we weren't allowed to show for a long time because Genencor paid for it and they didn't want it publicized, but they've now said we can talk about it. And it's very similar, but not at all, there are some significant differences in loops from the T. reesii enzyme, but in terms of the active site, they look the same. And so we have chosen 5 residues. So we already know a lot about this family. We were pretty certain this would be the catalytic acid, and indeed it is when we mutagenize it. We thought this residue was probably involved as a catalytic base, even though it's in totally the wrong place. In the model, you always see one on each side of the strand that's being cleaved, and this is on the same side as catalytic acid. And then serine, so basically there's a water molecule that's bound between these 2 residues that looks like it's the catalytic water. And that's why we think these 2 are important. And then these are 2 residues that are clearly involved in substrate binding, the hydrogen bond to the substrate. But there's a group that claims this is the catalytic base, and so we thought we would, recheck it with this family to see what we found. And so, we mutated each one of these residues to alanine and a couple of other residues that I won't be talking more about. And as you can see, with the D274, which I said should be the catalytic acid, it's basically dead on every substrate, and it clearly is the catalytic acid. And you can really prove a residue is the catalytic acid if you use paranitrophenyl cell biocide. It's got such a good leaving group that it doesn't need the catalytic acid. And the enzymes, if you've mutated the catalytic acid and used this substrate, you get wild-type activity on it. And that shows that everything about the enzyme is right. It's folded correctly, it's doing everything it should do, but it doesn't have a catalytic acid, and so it can't hydrolyze cellulose, which doesn't have a good leaving group and needs to have a protonation to get it to work. So that's a very clear result, and this, this enzyme, everybody who's mutated this residue in this one of these families all agree that it has the same result. Interestingly, as I say, people who work on T. reesii in my group feel this is the catalytic base, but the people who work on Pseudomonas fimi think that this residue is the catalytic base. So you can see that, look how much activity we have on the swollen cellulose substrates. It clearly isn't in our hands. And we can show that, if we look at oligosaccharide binding to these mutants, it's way down. And for some reason, they've never tested that, so we don't know if their mutants are low in activity. But they don't see any activity in their mutants, so that's why they think it's the catalytic base. Now, the really interesting result came when we looked at the actual products of hydrolysis, because if you notice this D226, even though it's inactive on BMC and swollen cellulose, it has slightly higher than wild type activity on CMC, and so we were really curious to look at the products that we got. And so when we did a mass spec analysis, we were absolutely astounded to find that whereas the main product from the wild-type enzyme is cell bios, this mutant doesn't make any cell bios, and I still don't understand that, because it does make cell triose, and it makes cell tetrose, and it makes cell pentose, cell hexose, and higher oligomers. So if you simply saw this data, you would say, we've opened up the active site cleft and converted an exocellulase into an endocellulase. But we know that isn't happening. We've done CD spectra on this enzyme. It's indistinguishable from the wild-type enzyme. There's no way that mutating, an exposed carboxyl group down in the bottom of the active site cleft, way away from where the two groups, the two loops are interacting to close the tunnel, could have opened up. And I actually think this is a fascinating mutant that I hope we'll be able to figure out how to study further, that has changed the ability of the enzyme to migrate along the substrate. So all the data on exocellulases says that if you kill their activity, they cannot move along the substrate. The processivity involves cleavage, the enzyme moving to fill the two empty subsites with the rest of the chain, moving backwards along the chain to fill the subsites. And that if it's dead, it just sits there. And that's been shown actually by atomic force microscopy with CBH1, and a lot of other indirect evidence says that's what happened. And so my belief is that the reason that this enzyme has high activity and gives totally different products is that unlike the wild type, which can only attack residues near the non-reducing end, this enzyme somehow can now move anywhere along the CMC strand, and anywhere it finds an unmodified residue, it can make a cleavage. And so even though it has to be cleaving more slowly than wild type because it's missing a key catalytic residue, it makes up for that by having so much more substrate that it can access. And so, because as I say, I know it has not become an endocellulase. It's become an enzyme that has the ability to cleave anywhere along the chain like an endocellulase, but it's, it's not, it won't reduce the viscosity rapidly. It doesn't, it doesn't behave like an endocellulase behaves at all. So, as I say, since we don't understand processivity, I think finding a single residue change that allows an enzyme now to move along the substrate, really, if we could figure out what's happened, could be really exciting. And so I mentioned about this argument over the catalytic base. And at this time, we don't know whether the C-Fimi enzyme uses a different mechanism than all the other members in the family, but obviously, what I believe. I'm afraid I think that something odd is happening with that enzyme, and that's why they're getting their result, not that it's really doing something different. But time will tell. Okay, so I mentioned the real test for catalytic base residue is azide rescue. And What's funny about family 6 enzymes till this time, we've never been able to see azide rescue until we tried it on this double mutant, where we have both the alanine and the serine mutated. We've done both single mutant enzymes, and they do not show azide rescue. In this case, it's a little odd because what happens is azide actually inhibits the wild type activity. And so what we see is an initial stimulation at lower concentrations of azide, and then we see the same type of inhibition that we see with the wild type enzyme. So it's very clearly that there is a very significant azide rescue. And so I think there's no question that these residues are participating in the catalytic base function, though I don't understand why we can't see the rescue with a single AS mutant enzyme. But this double mutant does have lower activity, and obviously the rescue activity is significant, but it's nowhere near back to the original total activity. All right, so I mentioned about our processive endoglucanase, and what's unique about this enzyme is that it has this extra family 3 CBM that's bound tightly to the catalytic domain, the family 9 catalytic domain, and it's bound in such a way that a cellulose strand bound into the active site cleft of the family 9 will go right along the binding surface of the CBM. And so basically, I believe that this CBM has become incorporated into the catalytic domain. And the reason that it's a processive endoglucanase is very simple, that it has an open active site. It can bind anywhere along the cellulose chain. Once it makes the initial cleavage, this chain will dissociate like any family 9 enzyme. There's nothing to hold it on. But because there's a very weak binding CBM holding this strand, it now has the option of going and filling the 4 empty step sites. And in fact, what it does is it cleaves cellotetrose processively from the non-reducing end of the chain. And we know that's what's going on because I've made a construct where I've deleted only this CBM from the enzyme, rejoined the family 2 CBM, and that enzyme is an absolutely classical family 9 endocellulase with 0 processivity in it. So it's clearly the presence of this domain that creates processivity. Now, we decided to do site-directed immunity, and this was the work of young Cellulase. So the work I showed you before about the family 6 and actually the antibody inhibition was all done by Thu Van Vong, who was a graduate student who just recently left my lab. And young Chao Li left my lab a little before him and did the work on the family 9. So he was mutating residues in this CBM, and he found this D513 residue. When he mutated it by itself, it actually had slightly better processivity than the original enzyme. And he'd also found this mutant over here in the catalytic domain had the same property. And surprisingly to us, when we put these 2 mutants together, instead of getting a better enzyme, we got a worse enzyme. And so this result suggests to us that the processivity and activity of this enzyme are critically dependent on the relative binding of substrate into this part of the molecule and into this, and that if this, we're not sure whether it binds more strongly or weakly. It's a weak enough binding CBM, it's hard to measure its binding affinity. But in any case, we've upset the balance, we think, by putting these 2 together. And that was certainly a surprising and somewhat interesting result. But what was fascinating that he did was, so he'd made a number of mutants that by themselves didn't seem to do very much to the enzyme. And we'd made a mutant a long time ago in which we deleted a loop, which is only present in processive endoglucanases in family 9. It's not present in other family 9 endocellulases. And the loop essentially forces this strain in the normal 9, it just goes straight through there. Because of this loop, it has to bend and come out here. And so we deleted that a long time ago, expecting maybe it'll change the products. Maybe the reason that this thing always gave cellotetrose was because it was like a bandsaw and you had a block there. You pushed your band in and cut it, and then push it in and cut it. But in fact, it didn't change. If you look at the properties of the mutant by itself, they're almost the same as wild type in every way. There's a slight increase in filter paper activity. That was the only thing. And I still don't know why young Chao decided to put these 2 types of mutants together, but it had an incredibly dramatic effect. And so here, and this is as a catalytic domain species. We don't have the family 2 CBM on them. So this one, where we put the two together, we got the worst result. But these three, all of them gave increased activity. And it's really unusual, because I've never before seen a mutant that increased CMC activity that didn't decrease activity on crystalline cellulose. And here's a mutant that somehow makes it better on all three substrates we tested. So we were really excited by this. And then, and there's just another experiment showing the data in a different way. So this is the bad mutant, this is the wild type, and these are two of the good mutants. And if you just look at this, you wouldn't say that's 2 or 3-fold more activity. But the way we measure activity is asking how much enzyme it takes to get a given amount of hydrolysis. And by that criterion, you need a third less of this enzyme than you do of the wild type to get a certain level of hydrolysis. But when we reconstructed this mutant, so we added back the family 2 CBM, which is a lot of work, because we had to express it in strip evidence to do this, and that's not easy to do. But Diana Irwin, who works with me, is very good at doing that, and she did it. We got the very disappointing result that even though the catalytic domain by itself, these mutants made the enzyme much better, when you added the CBM back, and the CBM species is 4 times as active with the wild-type enzyme as the catalytic domain. So to really get a useful enzyme for practical reasons, you need to have the CBM on it. Now they're worse. And so, it was incredible disappointment. I thought I made these beautiful cellulases, and when I put it all together to do it, it isn't. But if you think about it from a scientific viewpoint, I think this strongly suggests that there's something interesting going on between the CBM and the catalytic domain, because that's the only way I can figure out that these mutants somehow interfere, whatever interaction occurs between the CBM and catalytic domain, and that's why they're worse, even though by themselves they're better. And, so I can sort of imagine that, the CBM somehow helps to put the strand into this complicated structure, and these mutants somehow make the strand go in more easily. But then when you put the CBM on, which is making the strand go in better, it interferes with this other interaction that's important for the overall function. So, I'm trying to make a lemonade out of, make lemonade out of this lemon by saying that even though it's very disappointing from a practical viewpoint, it's actually interesting scientifically, which I really think it is, but we need to do more work to sort it out. So the final thing I want to tell you about is the work we're doing most directly on identifying residues that are involved in putting the strand into the active site of the enzyme. And we're doing that with cell 48A, and this is the work of Maxim Kozlovev, who's still working with me as a graduate student. And the reason we chose this enzyme is if you look at the active site tunnel, so you can actually see the hole in the active site where the strand goes in each of these structures. So these are 3 family 48 cellulases. They're 2 real structures. That's these 2. These are actually clostridial cell S and cell F. And this is our T-fusca sequence modeled on these structures. And we get a very good model because they're about 70% identical. So, it's really a very reliable model, I think. And what I find fascinating is if we look at what residues are within 10 angstroms of this tunnel entrance on the surface, which I think have to be the only residues that I can think of that on the catalytic domain could participate in putting a strand into that tunnel, every single residue on all 3 enzymes is a potential cellulose binding residue. Now, that isn't as impressive as it sounds, because in fact 2/3 of the residues are cellulose binding. But what is impressive is that aromatic residues are the most tightly binding cellulose binding residues, and I'll show you in the next slide. There's actually a 4-fold enrichment of aromatic residues in this region of the molecule compared to the whole surface of the enzyme. And there are lots of non-cellulose binding residues on the surface of each of these enzymes. The other thing that I think is interesting and fits with what I would say is my theory of why cell walls are so diverse is that even though all these residues on all three enzymes are potential cellulose binding residues, only a few of them are conserved. So there's a tryptophan, there's a serine along here. I guess it's this residue actually that's on all three. But most of them are different. And we'll find out when we do the site-directed mutagenesis, but my prediction is they're different not because they're not important, but because each of these enzymes may be specialized to break down a different type of plant cell wall, and therefore you need different residues for doing that job. And that's going to be fascinating to see if we can ever prove that. But it seems plausible to me. So this is the actual data. I just compared two of them, but the third is similar. So you can see this big enrichment in aromatic residues and none for both, all three proteins that are non-cellulose binding, even though they are significant fractions, those small fractions of the other surface residues. So we did mutate the tyrosine. It turns out, I mean, it's really fascinating that every single exocellulase, all three of them, you know, there are three different families, has a tryptophan at the entrance to its active site tunnel. And it's actually been shown a long time ago, Thierry was the first person to do it, to show that if you mutated the cell 6 exocellulase tryptophan, you did specifically inhibit crystalline cellulose activity. And we saw the same thing with cell 6b, though not quite as dramatic as what Tua saw, and we're seeing that here. So we actually mutated this residue both to an alanine and to a histidine, and we did the histidine because John Brady's molecular modeling suggested this residue would actually bind more tightly to the tryptophan, and so we thought it would be interesting to both remove the binding residue and make it more tight. And it's interesting that the tight binding actually makes the enzyme worse on all substrates than the alanine. So here you can see that on bacterial cellulose, this enzyme's only half as active as wild type, but on swollen cellulose, it's just as active. And on CMC, it's more active. So clearly, this residue is, in fact, the type of residue I've been looking for. It's really important for crystalline cellulose. I can't prove it's putting the strand in the active site, but it certainly seems likely that that's what it would be helping to do. So we've made another residue. There's a serine that I mentioned was conserved. It does nothing. So what's this? We're looking at the whole enzyme in this slide, you know, with the CBM on it, as well as the catalytic domain on swollen cellulose. And you can see that all three enzymes are very similar when we vary the amount of enzyme and look at the extent of hydrolysis. Oops, I went too far. And this is now the catalytic domain, and we're actually seeing a difference again. And I find that very interesting that we're actually in several different enzymes able to see differences in activity between the whole enzyme and the catalytic domain, which again, I think suggests some interaction between the CBM and the CD, but I don't know that it proves it. And so in this case, the serine's a little better than wild type. Tryptophan is worse. And okay, so let's see, I'll be careful which I'm looking at here. So there's now that was swollen cellulose. This is the BMCC CD. Oh, yeah, okay, so I went too far. Okay, so here is the mature enzyme on the crystalline cellulose. And again, there's the worst tryptophan mutant, the active there. And now here's, again, we're seeing the serine is better on both the BMC and the swollen cellulose, and the tryptophan is worse. So again, both residues are having some effect on this enzyme. And this is the CMC. And here you see, as I said, that you're seeing the better activity of the getting rid of the tryptophan. And for some reason, we see this regularly. Taking out tryptophans from the active site cleft or tunnel of cellulases almost always increases their activity on CMC. I assume because they allow modified residues to bind that couldn't bind before because there's more room, but I don't know that for a fact. So obviously, we're still just at the beginning of doing this mutagenesis, but I really think that, you know, having only 13 residues to mutate, we'll be able to get the whole picture for what is happening with the residues that bring the strand in for this enzyme. And I hope that'll allow us to figure out how the enzyme works, and then, if we're really ingenious, make a better enzyme. And lucky with combinations. So I'd be happy to answer any questions, and just should show the people. So I mentioned all the students who've done the research. Maxim, who's still in my lab, Thu was the first to leave before Maxim, and Young Chow before that. And Diana Irwin has worked with me for a very long time, more as a co-PI than as a technician, because she really put a real effort. And Mike Spezio did the structure I showed you of the Cell6A. And I've also worked with Ed Bayer in work I didn't talk about, where we've actually taken every one of the T. fusca cellulases, put a doctrine on it, and put it into one of Ed's designer cellulosomes. And the really interesting result is that when we tried to do it with the family 6 exocellulase, it didn't work. It was terrible in a cellulosome. But every other one worked fine when we put it in a cellulosome. So there really is something, though I don't know what it is, about that family 6 enzyme that just doesn't work in a cellulosome. John Brady's done a lot of molecular modeling with me, and Mike Himmel's funded a lot of my work, and we talk a lot about the enzyme work. And then Larry Walker is a colleague at Cornell who has done a lot to try and build up biomass research at Cornell, and he and I have worked together, and he's trying to do single molecules, and I'm hoping that some of my mutants, particularly that Cell6A with the altered processivity, we can get some interesting data if we do a single molecule with it. All right, so as I say, I'd be happy to answer any questions.
So you mentioned the potential problem of cellulosomes being too big to access the cellulose. What size limit do you think is too big?
Well, I would say that, I mean, we definitely see differences in accessibility between even 100,000 enzyme and a 30,000 enzyme, which is the range of what our individual enzymes are. And, cellulosomes are multi-million molecular weight particles, so they're way too big. I mean, the point is, if you actually look at something like Avicell, and you measure the surface area that is accessible to, say, a 40,000 molecular weight enzyme, more than 95% of that surface area is not on the particle surface. It's actually in these small pores. So, you really would expect that a cellulosome couldn't get in any of those pores. Now, I don't know, some of the enzymes can reach a little way down, and maybe they're so good at hydrolyzing, they just chew their way in, and it doesn't matter that the particle can't get in. Again, we really don't know what is going on there. But it always has seemed curious to me that, people who work on cellulosomes claim they're better than cellulases. But in my experience, that isn't true. They're the same. I get the same activity if I get a good cellulosome from somebody who knows how to make them, not me. You could just blame me as having bad cellulosomes. But, I mean, Ed sent me them, and basically, they're about as active as a T. reesii crude supernatant, a little better than the T. fusca supernatant. And I would argue that's actually very impressive given their very large size. And again, we have no idea why they're better, because it isn't that they have some unique set of enzymes. They really are the same sets, if anything more limited than the ones that are in the free mixtures. Yep.
So we see with the fungi the same thing you saw with the T. fusca enzymes that seem to work on hemicellulose or other polysaccharides. And then it has CBM. Did you test whether or not the CBM was essential for that ability to stimulate?
Oh, we didn't actually. That's a good idea. We didn't do that. No, I doubt if it would be in this case, but I bet it would really make a difference in a wall situation, where you really have harder to get at substrate. But it is an interesting point. We didn't actually do that. It is something we could do fairly easily and see if let's say maybe it would affect the amount of enzyme you had to add, for example. It might make a big difference there.
It's really, but we see like GH10 and 7, the same families I was looking at. Always have CBM and fungi produced by pure cellulose.
Yeah, that makes sense. I really think it's, again, a general, they're all facing the same challenge when they go after a cell wall. And xyloglucan is mostly in primary cell walls. It's not so common in secondary, but it's very abundant in certain primaries, and clearly. And in fact, again, one of the theories is that you really need it to prevent the cell wall from getting too rigid that the plant could never divide. If all the cellulose were crystalline and interacting with all the cellulose, this way you have a microfibril. Once you've got these xyloses sticking out, it can't bind to another cellulose chain, and so it definitely is very limiting for forming big crystalline cellulose structures.
the same sort of enrichment for tryptophans in there?
Well, I mean, the problem with the endos, the first enzyme I had a structure for was cell 6A. So I did a lot of mutagenesis of cell 6A, trying to find residues that would do the same thing as we did with this thing. And we never found any that had that property. And I don't know whether it's just because you have so many more to choose from and you don't know the right one to pick because you've got this huge long cleft with, a lot of residues at the top, slightly down in the thing, and you don't know. Or whether, in fact, the residues that are doing this function in the endo are actually also involved in catalysis. And so, even though they're doing both, and so when you mutate one, you knock out both functions and you can't tell. So I don't know what the story is yet there. But we definitely, I think we've mutated 30 residues on the surface of the tunnel without finding one that had that property in it. Yep.
So when you sort of deleted the exocellulase from the whole cocktail of the fusca, you saw a dramatic decrease in the percent degradation.
Yeah.
But when you deleted one of the more active processive endos, I think was it cell 9B, it was
almost no effect.
Almost no effect.
I know. That was a huge disappointment to me, and I still don't really understand it. But the fact of the matter is, when we do our synergistic experiments, make mixtures of pure enzymes, we can show, cell 9B individually will synergize with any cellulase we test. So it really has a different, my theory is that two enzymes synergize when they have different modes of attack on the sort of either the individual molecule or the fibril. And clearly this enzyme, because it can attack anywhere along the chain and then begin a processive cleavage, is really unique in what it can do. And we do see that it will synergize with either class of exo or with a regular endo very nicely. But in fact, if we make a mixture of our two exos and a good endo, we see a slight increase when we add this enzyme to it. But it's not a very dramatic increase because we already have a very high active mixture with the two exos and the good endo in it. So that would be my argument. But again, I don't really know, why the organism makes this enzyme. And it's actually interesting that, if you look at anaerobic bacteria, it makes tremendous sense to not have the exocellulase and to use the processive endo, because it turns out that cellotetrose gives 2 extra ATPs to the organism versus cellobios. And so because it only takes 1 ATP to transport any oligosaccharide, and with a phosphorylase you can get 2 extra ATPs from the cellotetrose when you hydrolyze it down. And so I think, that's why we don't see the family 6 exocellulase in anaerobic bacteria. According to Lee Lind, and I don't think Lee Lind is right very often, but here I do think he's right, is that, anaerobes, actually you have to produce so much protein to break down cellulose that if you only got the ATP you get from cellobios, you wouldn't be able to grow on cellulose as an anaerobe. And that you really need to have an average size of 4 sugars in your products to grow on cellulose. So it actually, if that whole idea is right, and I think it makes a lot of sense, it's really important for the anaerobe not to have that exocellulase and to use this other enzyme. And so that's probably why the enzyme involved, I would argue, and then T-fusca picked it up because it's slightly better than not having it, but it isn't really crucial for it from what we're seeing so far. And none of the fungi, it's never been found in a fungi for whatever reason. I'm sure they could make it. They have a few family 9 enzymes in them, so I don't see any reason why they couldn't produce it, but they haven't.
Well, then let's thank Professor